Hai pemain satu, siap hadapi tantangan ini?
Baru, Garnier Bright Complete 3-in-1 Anti-Acne. Dengan vitamin C dan salicylic acid, mampu melawan 99,9% bakteri penyebab jerawat. Terbukti secara klinis. Cukup satu produk, bersih, cerah, lawan jerawat. Beli sekarang.
Mana nih Manggia sama Hedi? Katanya ada di sini.
Ya, aman. Lah, udah pada datang nih.
Iya tuh Mang.
If you're an accredited investor, I have big news. In the last 12 months, I bought 5 major
One, two, breathe. 3, 4, and
If you're an accredited investor, I have big news. If you're an accredited investor, I have big news. In the last
Hello, my friends. I'm Hi from
Afternoon, Pak Asep.
Oh, halo Pak Arman.
Oh, Pak Asep.
Halo Dr. Ahmad.
How are you?
Fine. And you?
Yeah, I'm fine. You
Yes,
hear me
sir.
well?
Yeah. Now it's very clearly. So what time now in church?
Sorry?
What time now in church?
Now it's 8 or
Oh, 8.
15 minutes
Okay.
before 8 in the morning.
Okay. It's early or not?
Yeah, a little bit early.
Little bit, yeah. Okay.
Yeah, it's afternoon in your country now.
Yeah. Now is 12:46.
Ah, yeah.
Yeah, on summer we only different, I think 5, yeah. Yeah.
5 hours, yes.
Yeah.
Yes. Good.
If you want
And
to
how is
try,
the summer courses going?
yeah, it's now is 5 days. Now we come to
Uh-huh.
5. Yeah.
Yes.
Before every time is fine. Yeah.
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 Can you see my camera is open or not?
Okay, so it's still black. I cannot
Ah.
see your face.
Let me try again.
Yeah. But you can see mine.
Yeah, I can see you clearly,
Okay,
but
okay.
I already
Yeah.
opened the camera, but I don't know why it's not appear.
Oh, yeah, no problem. We still have enough time, but of
Yes.
course, participant want to see you.
Yeah, of
Actually.
course, I know.
Yeah.
Let me try it again. Now you see my profile picture only, right?
Yeah.
When I stop the camera, but when I start again the video, it
Uh-huh.
should appear like this.
I don't know. Zoom platform, it is common or not in Europe?
Yeah, Zoom is common. Yeah, we use it a lot.
Okay. Or how about the slide? Do you want to try? You want
Let
to
me try this share screening.
Yeah.
Is it now clear?
Yeah. I can see, but maybe now it's full screen. Yeah.
Yes.
Yeah, it's nice.
And it's working well?
Yeah, it's working well. Yeah.
Okay.
It's nice.
I sent you also a PDF about this presentation on the email just a few minutes ago.
Yeah.
Also
I
check this.
received already.
Yeah. So let me check this video. I open, for example, on Skype now. It works well. The camera is working, and I can see myself.
Yeah. Really?
But I don't know why in Zoom it's not working. Maybe you
Yeah.
have to allow something from your side or what?
Okay, I don't know. Maybe I can ask to the committee, Parman, how to show the picture actually.
Maybe there is an option to allow for also video, not only mic, but also for video.
Oh, you want to share also video, yeah?
No, I mean the camera, just to see me.
The camera. Okay.
Yeah,
Okay.
maybe you have to allow also this option. I don't know in Zoom.
So I don't know. Maybe I can ask. Okay, Pak Arman atau Pak Jimas, they have any like menu or other maybe application who can open? Oh, okay.
I went already to the video setting and I have option of camera and I select this HD. Or maybe this.
So you already select the what is the setting, yeah? And then you choose the camera.
Yes.
Yes.
I
that
have
is-
the camera option, the HP HD camera, and I select HD.
Yeah, now it's working, but
working.
it is
Yeah,
yeah.
Yeah,
Good.
now it's working.
Yeah, yeah. So your background is planet.
Yeah, yes. So you can see now it's clear.
Okay.
Good.
Yeah. It's clear now.
Oh, great. Yeah, there was, I think, option that I have to remove it. So now it's selecting right.
Okay.
Let's try again the sharing screen. Now you can see it in full screen. Yeah.
In full screen. Yeah.
Great.
Good.
No,
So-
good. Great.
You can take a copy before you talk. At least 100 minutes with discussion.
Yeah.
Hello.
So it's 100 minutes, around 100 minutes.
Yeah. So I introduce who will moderate your talk today, Pak Baihaki, Mr. Baihaki. Maybe so you can say something. This is Associate Professor Dr. Ahmad Kiat already here.
Yeah.
And we communicate how to, because before cannot show the camera, but now you saw already. Yeah.
Okay,
So
thank
hello,
you.
Dr. Baihaki.
Hello,
Nice to
Professor
see you.
Ahmed. How are you?
Fine. What about you? And everything is good? Well,
Yes.
great. So how the lecture will be managed, or what do you expect the management?
Yeah, this is more or less 1 hour and a half.
Okay.
And then 1 hour is your presentation, and then 30 minutes for discussion.
Okay.
Okay, so and then yeah, that's all I think, yeah, for our class for today.
Okay, great. And during the presentation, do you allow questions, or the question will be at the end only?
Yeah, I think if the participant has any question, so maybe they just take it or keep it by their mind, and then afterward
okay.
maybe we will
After
collect
the
it.
end of the presentation.
Okay.
Okay.
Yeah.
Yeah.
Normally
Okay,
we
fine.
collect the question, and then in the end maybe so we can roughly to answer.
Yeah.
Or maybe sometimes they also raise hand if they want to ask directly. So maybe it's also good.
Okay. In all cases, it's fine for me. No problem. If someone would like to stop me and to ask
Okay.
in the middle, it's fine.
Okay.
If someone would like to keep the question at the end, it's okay. No problem.
Okay, but yeah, I think it's up to you. I think it's better.
Yeah,
Which
for
one
me,
is better?
both options are okay for me. No problem.
Okay. Good.
Yeah. Now we waiting maybe little bit, Ahmed, because in here is Friday time.
So we still have 5 minutes, or?
Yeah, we still have
Normally
5
we
minutes,
wait,
more or
yeah.
less.
After 5 minutes, normally they increase until 100 participants. So normally
okay.
stay on time.
Okay.
On time in 1 o'clock.
Yeah.
I think still in your place is still in the morning, yeah.
Yeah, here it's almost 8 a.m. in the morning, so it's very early.
But summer, I think is 4 o'clock also already.
Yeah, it's already. Yeah, the
you can see
is early,
yes.
but we usually start working from 8, or I usually come here at 8 or 8:30 maximum.
Okay.
So that's normal
Yeah.
time for working. Yeah.
Yeah. Sorry, this is not good for you. Actually, it's good
No,
for
it's
vacation,
fine.
for holiday.
I was just already in holiday. I just arrived last week, so
Okay.
so it was
So
my holiday. This time I already finished.
okay. So to where is your holiday?
I was
Still
in
in Europe?
Egypt.
Oh, back to Egypt.
Yeah,
Okay. Okay. Okay.
just
Okay.
to visit my family and my
Okay.
friends, and then for 1 month,
Yeah.
and
Yeah.
then
Yeah.
I
Yeah.
come back here to Czech Republic.
Okay.
Yeah.
Okay. Okay. So you already, I mean, familiar with this situation. Yeah.
Yes, yes. Don't worry.
Yeah. Fly to Canada. Fly to.
You
Yeah.
remember this? Yes. Yes.
Okay. Yeah. Actually, Czech is close to Germany. Yeah.
Yeah, it's very close. It's in the border actually. So it's the same maybe weather, the same very close, and so I did the same time zone also. So we don't feel the difference. It's like you are in Germany. Not a big difference. Yeah.
So how about the situation now? When I saw the Euro, is people already coming to the stadium?
Yes.
It is without the mask. Mask?
Actually, yes, because of this vaccination process. So they are start to vaccinate a lot of people, and they allow people who are already vaccinated to go without using the mask and so on. But still, we have also some doubts or afraid about the new strains of the virus, as it's appeared this Delta strain and also Lambda strain. So maybe the number start to be increased again, number of cases of infected people. So hopefully we will not face the fourth wave of infections. We are waiting. Yeah.
Yeah.
And how is the situation in Indonesia?
Yeah, now in Indonesia is increased. Yeah, increase to become increase, and then we have a restriction. Yeah, restriction. Yeah, I think from where Pak Bay? From 3 until 20. Yeah, 20.
Yeah, until 20 of July.
I don't know. Maybe there is
a restriction for people
renew
to
or not.
cross the border.
Yeah.
That's why you decide to do this summer course online, virtually, or normally it's doing like this.
This is the fifth of summer course actually, but since last year we use or we conduct this summer course to become online. Maybe yeah, because we are not sure actually from the beginning of this year. We decide to conduct this online summer course.
Okay.
Yeah, actually
nowadays,
this also
most of the conferences and seminars are now by using online systems. So no one is going physical to meet each other because of this COVID-19. So here in Europe, mostly all meetings are online.
Yeah. So this is maybe also we have also advantage because we can coverage more participant, more people coming from different country. Yeah,
right.
Otherwise maybe
more easier. Yes.
Yeah. But yeah, maybe we cannot see face to face.
Yeah, that's a problem. So normally when you see face to face is much better, of course, and the communication is much easier.
We have a lot of experience to maybe to know each other, and then to link between like that.
And I hope the topic that I suggest that I will present today will be also of interest of the participants. So I don't know if they are interesting or not in this topic, but hopefully they will get the benefits also from that.
Yeah, because this is mostly animal production. So so I think they will really, because we need also some kind of this advanced technology. So maybe we can share like that.
Great.
So this university is life science.
I am here in the Institute of Animal Physiology and Genetics. It's related to the Czech Academy of Science. So it's one of the institutes of the Academy of Science. And they are focused mainly on animal physiology and genetics research related topics.
Yeah, it's in line, yeah,
and it's like the one that we have in Germany in Bonn. So the same field of study of this embryology and embryogenomics and the IVF stuff and things. We work also with bovine and porcine species. So it's looks like similar to what we did in Bonn.
Okay, good. So you have more advantage, yeah? Can speak Deutsch and also now Czech.
Yeah, Czech, but the Czech is really hard to to
Really?
learn actually. Yeah,
It's
so I
otherwise
learned very few
Germany
words.
and Czech is close, but the language is totally different.
Totally different. Yeah, so Czech is more related to Russian language, not to Deutsch. So
Okay.
so it's a big difference between Deutsch and Czech. So it's hard to learn also both.
And they also like they have a different, I mean, alphabet like a Cyrillic, yeah. Russia is like a
Different what?
alphabet, how to write.
Yes, they have. It's like also the English and Deutsch, but they have some special characters also with some signs. So this with different pronunciation and a little bit different, but the characters are the same like English and Deutsch, not like Russian.
It's already 1 o'clock, Pak Asep.
Yeah, please, bye. Maybe we can wait also for others. Yeah, bye. So the time now is yours.
Okay, thank you very much, Pak Prof. Asep. Good afternoon, everyone. Yeah, today, as we did for yesterday, we will have two lecturer that will share their knowledge and their experience as well to us. And now, yeah, as you see in your screen, yeah, we have Professor Ahmed Gad from Czech University. Yeah, I hope everyone is already here. And as I mentioned before, yeah, during this seminar or this session, maybe if you have any question, yeah, for participant, you just keep in your mind, or maybe you can write down in our chat room, and later on we will collect it. And in the last session, I mean in the discussion session for 30 minutes, yeah, we will have any question and answer. Okay, Prof. Ahmed, yeah, again, thank you very much for joining with us. And before I give your time for your presentation, let me share to the audience about your curriculum vitae. Okay, let me introduce Associate Professor Ahmed Yahya Gad. Yeah, this comes from Department of Animal Production, Faculty of Agriculture, Cairo University. And then if you have, yeah, maybe later on, if you have any, yeah, folder of discussion, you can write down his email like here, yeah. And yeah, the education of Professor Ahmed, yeah, for BSc and MSc program, he finished in Cairo University in Egypt. And then for PhD program, he conducted in University of Bonn, Germany. And yeah, he has many work experience, as you can see in your screen, from 2017 until now. He is postdoctoral researcher in Institute of Animal Physiology and Genetics in Czech Academy of Science, Lebíčov. I hope I do not mistake.
Yeah, this is the name of the city.
Okay. Okay, Czech Republic. And he is also as associate professor in Faculty of Agriculture, Cairo University in Egypt. And yeah, this is the publication of Professor Ahmed. You can see maybe you can check it in Google Scholar page. And as you can see here in this cycle, yeah, his age index is 14. I think it's very high. And also for the document and so on is very, there is a lot of document from him. Yeah, the document, as you can see in the screen, yeah, until now in Scopus 31 and with total citation 677. yeah, very high, I think. And this is the award and also fellowship that Professor Ahmed have. And you can see also here, yeah, many of them as a first prize, and first prize. So I think there are a lot of things that I mean the awards and also fellowship that get of Professor Ahmed. Okay, and this is the membership of scientific societies that Professor Ahmed included. Yeah, for example, in International Embryo Transfer Society in USA, in Egypt itself for Egyptian Society of Animal Production, and also Egyptian Poultry Science Association. Okay, no further ado. This is the topic for from the Associate Professor Ahmed Kat about extracellular vesicle new biomarker in livestock production. So let me stop my screen. And now, yeah, the screen is yours, Prof. Please.
Thank you so much for this nice introduction. So I will start to share my screen with you. I hope that you already now can see the slide.
Yeah.
It's clear and full screen. Okay.
Yeah.
Great. So thank you again for this introduction, and thank you very much for the invitation for me to give lecture in the summer course in this year. I know it's become like online session, not physical, but hope that all the participants could get also benefits from the lectures. So the lecture of today, the topic is, as mentioned, extracellular vesicles and new biomarkers in livestock production. So I hope that I think it's a new field of science now, and it's appeared recently, especially in animal production science, the topic of extracellular vesicles. So we will know today what is the meaning or what is the definition of extracellular vesicles, the structure and function. Then I will talk about the methods of isolation and characterization of these extracellular vesicles, and we will call it EVs, just to make it simple. And then I will talk about some applications of these EVs in livestock production. It's still the application on the research level, but I think recently it will come to the commercial or to the farm very soon. And then I will end up the presentation with conclusions and the challenges. So first, let's see what is this. The question is, what are extracellular vesicles? So as you can see here in the screen, this is the graph of the common graph of extracellular vesicles. It is a nanocyte lipid bilayer membrane that are secreted from all kind of cells in all organisms or in multicellular organism and also in bacteria. So it is a very small particles, nanocytes, and as you see here in the graph, so they have a lipid bilayer membrane, so two or double layers of lipids. And the most important thing, that is what is inside. So inside this nanoparticles, you can see different molecules or different bioactive compounds like DNA, messenger RNA, microRNA, proteins, and lipids. So this is the component. And the most interesting point also that these nano-sized particles or EVs could be found in mostly all body fluids, whatever the fluids in the body, for example, in the blood, in the urine, in the seminal plasma, in the oviductal fluids, all kind of fluids in the body, you can find these small particles in these fluids because they are secreted from the cells. And here this photo, if you can see this photo from the electron microscope. So this is what the EVs look like under the electron microscope. So you can see here small size. So this is, for example, let me change this word. Yeah, so the bar, for example, here is 200 nanometers. So this is black bar is 200 nanometers. So they are smaller maybe than 200 nanometer in diameter, but they are look like this shape. So you have to memorize this photo. So Then coming, what is the function? What these EVs doing? So in the beginning, from long time ago, so people consider these EVs like a garbage bags or cell waste. So like the cell secrete its waste by using these particles. Then, but this was not the right thinking, but after that, and recently, it was discovered that these particles has very important role in intracellular communication. So they play a very important role in communicating cell by cell during several conditions, under several conditions, like example, physiological condition or pathological conditions. So in both cases, there is a communication between the cells under these conditions, and these EVs play important role in this communication. So the new definition of these extracellular vesicles are cell-derived nanoparticles that can carry message from one cell to another. So this is very important to also know that they carry messages. If you look to this timeline or the history of discovery of EVs, so it's not recent as a discovery of, as I said, in function, but as a structure, it's discovered a long time ago. So it's starting from 1940s. So but it was discovered just as a dust, as I told, so for example, platelet dust or procoagulant derived particles. They don't know what is the function or what they are doing, but they found it under microscope. And in 1969, also it's called like matrix vesicles. So they saw some vesicles under microscope, but and it looks like matrix, but they don't know what they are doing. And the first definition or the first report of the word extracellular vesicles appeared in 1971. So when they saw under electron microscope, and they called it EVs or extracellular vesicles. And after that, in 1980s, for example, the first word of exosome. Exosome also is another word that can be used for these particles, but as we will see later, what is the meaning of exosomes? And it's going on until in 1996, for example, it start to have some information about function. So they found that there is a change in the exosome secretion under the immune response of the body or the cells. So they suggest that these particles could play a role in immunity. And the most important discovery was in 2006, 2007. So I consider this year is the real start of the science or the technology of extracellular vesicles, when the two different labs or two different researchers groups discovered that these exosomes or extracellular vesicles contains biological molecules like RNA, like proteins inside the cell. I don't know, do you see the mouse that I'm moving now or not?
Yeah, we see that.
Okay, great. So in this year, 2006, 2007, this was the real discovery of the function of this, because they discovered that there is something inside. It's not dust, it's not particles, empty particles, but it's containing something inside and something important like RNA, like protein, like DNA. After that, the research is going, and there was the Nobel Prize in 2013 about the discovery of the regulatory mechanism of this vascular transport system, and this was won the Nobel Prize on that year. And since then, there is a lot of research going on, and clinical trials also in recent years. And if you can see now in PubMed, there is a lot of papers coming out, or research papers coming out with the work related to extracellular vesicles and exosomes. If we look to this figure, for example, from PubMed recently, if you just write extracellular vesicles, you will see like thousands of papers, but it starts really from, for example, 2009 to be increased like 464 papers per year. But now, last year, we have like 5,000 and more papers in 1 year. So, and this number is increasing dramatically year by year, because people in all fields of science start to see that there is important role of this extracellular vesicle, either in medical science, in basic science, whatever the science. So they play a role in a lot of fields of research. So these extracellular vesicles can be classified according to two main criteria. The first criteria is the size, and the second criteria is the biogenesis. So if we look to the size, for example, so these extracellular vesicles can be classified in three main groups. The first group, as I told you, these exosomes, and this is the smallest type of extracellular vesicles. The diameter of the particles is around 30 to 100 nanometer. Then coming the second time is the microvesicles. Microvesicles is a little bit larger, and the diameter is from 100 to 1000 nanometer. And the last type is the apoptotic bodies. Apoptotic bodies is around 1 to 5 micrometers, so it's a little bit larger. So this is the main classification of EVs based on the size. And if you look to the photo under electron microscope, you can easily recognize the difference in the size. For example, this looks like exosome. It's a smaller one. As I said, it's from 30 to 100 nanometer, for example. Microvesicles is a little bit larger. It can reach until 1000 nanometer. But apoptotic bodies is much, much larger. Is around, can reach like 4000 or 5000 nanometer. So it's a big difference between them in the size, but not only in the size, but also in the biogenesis, how they are created inside the cells. So if we look to the first picture, for example, exosomes. Exosomes are created or formed within the endosomal network from inside the cell, and then merge with the plasma membrane and then released outside. However, the microvesicles is produced by outward budding. It's just budding from the plasma membrane, as you can see here in the photo, just extended or budding from the plasma membrane and then released inside or in the circulatory system or in the fluids. However, apoptotic bodies is usually related to the apoptosis of the cells. When the cells going to die or to apoptosis, so they are fragmented into small pieces, and this fragmentation bodies is called apoptotic bodies. That's why most of the research you will find they are interested in the first two types, exosomes and microvesicles, because these are natural and in healthy cells they are produced under different conditions. However, apoptotic bodies is produced only in the case of apoptosis or cell die. Again, you can see here the different biogenesis. So as I told here, microvesicles are budding from the plasma membrane. However, exosomes are created inside with the endosomal network and then fuse with the plasma membrane and release. But apoptotic bodies from apoptotic cells is like a fragmentation of the cell. Here also you can see in a little bit in more details about especially the exosomes. So exosomes start with this endocytosis process. So the cell endocytoses the outside, for example, or from the surrounding area of these cells, and then creating something called early endosome. This early endosome going to create what's called late endosomes, and they are trafficking inside the cell until it's creating something bigger called multivesicle bodies containing this small exosome inside. These multivesicle bodies can go through two different ways. The first way is to go and fuse with the plasma membrane, and then the exosomes will be released outside. Or the second way is to fuse with the lysosomes and it will be degraded. But no one knows, and still we don't know exactly what makes this multivesicle body going in this way or going in this way. So it's not known exactly how it is working. So this is what people would like to see. I just would like to use this spotlight. Yes. Then, as I told you, so they are fusing with the plasma membrane, and then is going, the exosomes are released in the circulatory system or in the body fluid. However, the microvesicles, as I said, they are just starting as outward buddings from the plasma membrane, and then they are released outside. That's why when we look to the structure of this extracellular vesicles, you can see there are some specific markers inside the membrane, as I show you this photo before. So you can see there is some what's called tetraspanin, like CD63, CD9, CD81. They are specific for exosomes and for extracellular vesicles. So they are markers on the membrane of these vesicles, or some other proteins like major histocompatibility complexes. They are also one of the marker proteins are embedded in the plasma or in the lipid membrane of this particle. This is very important because we will use this later to show how we can characterize or isolate these exosomes based on these specific protein markers. So this table just showing again or summarize the difference between the three main types, these microparticles or microvesicles, exosomes, apoptotic bodies. As I say, that the origin from plasma membrane, this origin from intracellular endoplasm or endocytosis, and apoptotic body is just fragmentation, so it's not determined. The size also, as you can see, from 50 to 100, here from 100 to 1000, and here from 1000 to 4000. The shape, especially exosomes, are very recognized by this cup shape. So it's like a cup, and I will show you also some photos of that. But the microvesicles and apoptotic bodies is irregular shape. This is shedding from plasma membrane, this is from exocytosis, and this is apoptosis. So as I mentioned in the beginning, that these microvesicles or exosomes or extracellular vesicles could be found in any body fluid. So as we are interested in animal reproduction, so we are interested in body fluids that related to reproduction. For example, of course, blood is also one of the interesting body fluids, so we can isolate exosomes or extracellular vesicles from blood. Milk also can be used, and all others fluids related to reproduction system, like uterine fluids, follicular fluids, oviductal fluids, and also it could be used from urine. And of course, from male, we can isolate it from seminal plasma. So sorry. So that's why, and also not actually only the from body fluids, but also from in vitro culture. So if we have cells or embryos are cultured in vitro, so these cells release some extracellular vesicles in the culture media and can be used from this culture media or can be isolated from this culture media. So any kind of fluids related to biological materials, you can find some EVs inside. And there are different methods for isolation. For example, so different methods of isolation. One of the most common method is this differential centrifugation, and it used ultracentrifugation because it's very nano-sized and small particles, so it needs very high speed of centrifugation, which reach around 100,000 xg, and for longer time, for around 70 minutes, just to isolate these exosomes. There are also another type of ultracentrifugation, but it comes with the density gradient ultracentrifugation, and in this case, you isolate the particles based on its density and also based on its size, so it's a little bit more accurate. There is also methods for size exclusion chromatography, so it's used coulombs, and these coulombs can capture some particles and release other particles based on the size, so you can also take out your particles from there. And a lot of other methods, I will not go in details, but there is a lot of methods now in the commercial fields, and you can find a lot of kits in the market to isolate these exosomes or EVs from any kind of body fluids. And of course, the difference in body fluids can also affect the method of isolation. So there is some kits specific for blood, some kits specific for other fluids, or for milk, or for plasma, seminal plasma, and so on. For each method, there is some advantage and disadvantage. So I will not also go in details for each, but for example, if you see the differential centrifugation or ultracentrifugation, this is one of the most used methods. But the problem of it, it's time consuming and also not specific. So some other particles like lipoproteins, like other small proteins aggregates can be also isolated or sediments with the sample. So it's not that accurate enough. However, for example, some other methods like density gradient is a little bit high purity more than the differential centrifugation. Some other methods like precipitation, for example, it's simple and fast and easy and maybe also cheaper, but it's also low specificity. So you cannot get a pure EVs from that. So each one has to consider which method can be used, because until now, there is no standard method for isolation. As you can see, each method has its advantage and disadvantage. So you have to manage, and also some methods depend on the volume of the sample. So for example, this size exclusion chromatography is always for small volume of samples, not for large volume. So if you have large amount of samples, you cannot use this method, and so on. So each one has to consider which method is better to be used for isolation of EVs from the body fluids. After isolations, coming the characterization. So characterization here means that we need to see what we isolate. So there are also several methods for characterization. The most important is this electron microscope, just to see that the vesicles that you isolate is the right one or not. Also, there is very important method of exosome tracking or nano tracking analysis, or this dynamic scattering. This is to show you the diameter of particles that you isolate and the concentration of these particles, as I will show you also the results coming out. Also, there is some protein analysis methods like this Bradford protein assay or Western blotting. Also, this is important to make some quantification and also to see the specificity. Did you isolate a real exosomes, or you isolate other things and you have contamination with other things, and so on. So there are several methods for characterization. Each one should also follow these methods. So if we look, for example, to this electron microscope, as I showed you this photo before, so this is a different size of extracellular vesicles like exosomes, multivesicles, and apoptotic bodies. So this is what you have to see under a microscope if you isolate. Or if you isolate only exosomes, you will see this shape. As I told you, exosomes are very simple, smaller, and also characterized by this cup shape. Cup shape, it's like a cup, and this double layer of membranes can create this cup shape. So if you see this under microscope, it's very clear that you isolate a real exosomes without any contaminants without it. So this is one of the characterization methods, this electron microscope. The second method, as I told you, is nanosites, and this is an example of real reports coming out from nanosites. The most important thing you want to see is this, the mean of the diameters of your particles. So here we have like 100, almost 30 nanometer, which is in the right diameters of extracellular vesicles or exosomes and the microvesicles. And also it's important to see the concentration, how many particles you have per ml of the body fluid. So here, for example, we have almost 2 to the 10 power 11 particles per ml. This is usually, you can see this high number in blood, for example, or in floccular fluids. You can get this high number of particles, billions of particles in 1 ml of this body fluid. So this is also important to see. It means that there is important function of these particles. It's not rare or not a small amounts of particles. No, it is a very huge number of particles in 1 ml of the body fluid. And the last method of characterization is the protein Western blot. If you remember the photos that I told, that there's some markers inside or embedded in the membrane of these particles like CD63, CD9, this is important to see if these markers are there. So you have antibodies specific for this tetraspanase proteins, and then you run Western blot and see from the cells and from the exosomes. You will have to find this band. It means you have really exosomes in your sample. Some other markers very specific only for the type of EVs, which is called exosomes. So like Alex, like TSG 101, they are specific only for exosomes. So you have to find the band if you really have exosomes in your sample. And the negative marker, usually we use any proteins related to the cells, like clandestine or like mitochondrial related proteins. This should not appear in your exosome isolation. If it's appear in your exosome isolation, it means you have a contamination with the cells. So you have to repeat the isolation. So if you have a pure isolation from exosomes, proteins from the cells should not be appear, but it should appear only in the cell samples, not in the exosome samples. So by using these three methods of characterization, you can say that I really isolate enough amount and isolate pure amount of EVs or exosomes, and then you can go further for other analysis. So then coming the next question, how these extracellular vesicles are taken by the target cells or by the recipient cells. So they are released, as I said, from the parent cells or the donor cells. So they are released in the body fluids, in the blood or whatever, but they are reach other cells like these target cells. So how these exosomes or EVs can affect the function of these target cells. There are different scenarios, and still now people work about to understand how they are, what is the specificity of this targeting. But one of the scenario is this receptor ligand interaction. So like interaction between the receptors in the EVs and the receptors of the cell. All these kind of lines describe different interaction between receptors. And once these receptors are interacting together, maybe there is effect on the recipient cells or in the target cells can be happening. This is one scenario. Another scenario is a direct membrane fusion. So these EVs could fuse with the membrane of the target cell and then release its content inside the cell and then affect its function. Another scenario is this phagocytosis process. So the target cell can phagocytose these microvesicles or EVs and then can digest it inside and release its content and doing the effect. And the last is some specific pathways related to endocytosis could be also one. So until now, we don't know exactly what's happening. Maybe all four scenarios are happen or pathways are happening in different cells. Maybe only one is happening. Until now, the research is going on to understand how these EVs could be uptaken by the target cells. Then coming the main question here in this presentation, why we are interested in EVs. So why the researcher in animal science, for example, or animal production, livestock production are interested in EVs. As we said that EVs contains and deliver cargo. Cargo, it means they have message, they have molecules inside, like, as I said, microRNA, messenger RNA, proteins, DNA, and it's not random, it's selected and it's related to each type of cells. And also they are sent as a message to another type of cells. So it is very important to understand this communication. Also, the most important thing, it's not a static system, it is dynamic system. So it is deregulated under different physiological conditions. So for example, under heat stress, you will find different types of exosomes with different contents. Under normal condition, totally different. Under disease condition, different. So it's not a static system, it's dynamic system and it's changing based on the physiological condition. Therefore, it can be used easily as a biomarkers or diagnostic tools for disease, for example. And this is very important in our field of animal science to find biomarkers for any kind of physiological condition or for any disease to diagnose this disease. So you can use this, and because this is very easy to find, because as I said, it's in all body fluids. So you can simply isolate it from blood and you can diagnose the disease or the physiological condition based on this isolation. And the most important also thing that can be used as potential therapeutic tools. So it could be used as a therapeutic or as a drugs in the future. Now researchers start to use these AVs as drugs, so they can also load these EVs with specific medicines, with specific molecules, and then inject it to the animals and then see the effect later. So that's why we are interested in these EVs as a biomarkers, as diagnosis tools, or as a therapeutic tools. And the fields are different. So as I told you, the beginning is to isolate and to characterize. This is the first part. And people are working on this, how to improve the isolation methods, how to improve the characterization methods. And then coming after that, the analysis. So our researchers are interested, some people are interested in RNA analysis, some are in protein analysis, some are in lipid analysis or metabolite analysis, or maybe also DNA analysis. As I told you, all these are inside these particles. So each lab or each research groups are interested in one of these different fields. So, for example, in our lab, we are very interested in microRNA analysis inside these particles. So there is a lot of microRNAs are released inside the EVs, and of course they have a function. So we'd like to understand what they are doing and why we are interested in microRNA. MicroRNA, maybe you already know that they are a small non-coding RNA that are transcribed inside the nucleus in the cells, and they are released outside in the cytoplasm, mature until they reach the maturation here as a mature microRNA, and they play a role in different functions. So, for example, they can target a specific messenger RNA and they cleave it. They can make a translation repression or messenger RNA adenylation. So, as you can see here, this microRNA, the green one, can specifically target one messenger RNA. In this case, it will make a translation block, so the protein will not be synthesized or not be translated, or can be make deadenylation of this messenger RNA and degradation of this messenger RNA. So in this case, the expression of the genes will be stopped. So microRNA is very important because they're regulating the gene expression. And the most important also that one microRNA could target hundreds of genes. So if you know one microRNA, you can see that it could possibly target a lot of genes and stop this expression or stop the translation of this gene. And the other, that one gene could be also targeted by several microRNA. So it is a very important regulatory mechanism that regulate the gene expression, and of course, it will regulate the function of the cells. So now, after we finish the first part, it means that we understand what is EVs, what is functions, what is the structure, why we are interested in it. Now I will show you some application in livestock production. As I told you, this application is still in the research field or still in the lab level, but some of them start to go outside in the farms and the field, but not as much as we expecting now. But I think in the near future, a lot of application will come out in the field for diagnosis or for biomarkers or even for therapeutics tools. So as I told you, we are interested here in reproductive physiology. So you can see a lot of applications in reproduction. So if we look, for example, for oocytes in the ovaries. So it's the oocytes, when it's in the ovary, in the flocculars, there is a lot of extracellular vesicles. These small blue dots are released in the floccular fluids. And they found that they play a role in flocculogenesis, in oocyte maturation, in oocyte competence, and so on. So each cell inside the follicle releases different types of extracellular vesicles with different contents. And after that, when the oocyte is ovulated and going in the oviduct, there is also a function or some EVs released from the oviductal cells, and also some are released from the oocyte itself. So it's a kind of communication, bidirectional communication between the oocytes and the cells of the oviduct. So this is also related to interaction between oocytes and oviduct. When also the fertilization occur in the oviduct, so this fertilization EVs play a role in also increasing the sperm viability, motility, to inhibit the polyspermy, to help the sperm to go to the capacitation, and all steps of physiology or reproductive physiological steps related to oocytes and the sperm are occur here is also managed by the release of these EVs. And so on, when this embryo is fertilized and then start to cleave, so the communication between the embryo and the oviductal cells, or after that with the uterus, is also managed by the release of extracellular vesicles. As you can see here, for example, the uterus release some EVs, which has totally different function than the EVs released by the oviduct. So here, for example, EVs in the uterus inhibit the acrosome reaction and capacitation of the sperm, for example. And here it's increase the capacitation. So it's a kind of different function based on the position of the physiological events happening. So we need to understand what is this kind of communication happen, how can we use it as a markers, how can we use it for therapeutical process. not only in the in vivo condition, but also in vitro condition. So for example, in the in vitro condition, we collect the gametes from the ovaries, or we collect semen and do the fertilization in the lab, and in the petri dish we see the embryos are developed. So all these steps also have showing that the EVs play a role. So for example, in embryo culture, the embryos release these EVs in the culture media. So what if we can add these EVs to the culture media to improve the development of the blastocyst or the embryos? And also after that, the transfer and the uterine interaction, also they have a role on that. So I will show you some examples, for example, from follicular fluids. As I told you, this, if we consider this is the follicle, and this is a different type of cells, Zika cells, granulosa cells, cumulus cells, and this is the oocyte. So there is a communication between these cells, and they are affecting each other. And at the end, they affect the oocyte competence. If this oocyte could release normally and could produce a normal embryo, or this oocyte will degrade it and not produce the right embryo. So simply, if we just collect the follicular fluids from here and try to understand what is this kind of EVs, what is their contents from protein or microRNA or lipids, whatever, so we can understand this communication, or we can use it as a biomarker. As I will show also here, for oocyte selection, for example, this is one of the main applications that we are working in. Oocyte selection, to select the right oocyte to be fertilized or to transfer it later as an embryo. So there is some morphological evaluation, like to see the cumulus cells as here, or after maturation to see the polar body. But all this morphological evaluation is subjective, and it's also different from person to person, and it's not that accurate. Some other method using staining to stain the oocytes with, like, for example, PCB stain. And the stained oocyte, it means high quality. Unstained oocytes mean low quality. But in this case, also it's a kind of invasive method, so it can affect the quality or competence of the oocytes because you add chemicals to the media. So that's why we need to have accurate methods for separation or for sorting or for selection of oocytes, but in the same time, we don't need it as objective or subjective method or invasive method. So we need some methods for without touching the oocyte. So that's why we think about use of follicular fluids. So follicular fluids which are surrounding the oocyte. So in this experiment, we isolate each oocyte individually, and we collect the follicular fluids around this oocyte. And in the same time, we stain the oocyte with another stain called the Lissamine green, just to see bad quality oocytes and good quality oocytes. And then we take the follicular fluids around these oocytes, and we do this EVs isolation and then microRNA sequencing. So I will show you some results from this experiment. As you can see here, we characterize, as I said, the three methods of characterization with Western blot to be sure that we isolate the right EVs. We saw them under electron microscope, as I show you also this nice shape of the exosomes, the cup shape. And we found that the good quality follicular fluids or related to the oocyte quality, showing a little bit higher number of EVs in the ml compared to the low quality. And both showing the same size or the same diameter, like 130 or 135 nanometer as an average. So this is just a characterization. After that, we compare the microRNA inside these particles. It was very interesting to see that the good quality and the bad quality, they have different expression of microRNAs. They have unique microRNAs expressed only in the bad quality follicular fluids or follicular fluids coming from bad quality oocytes, and also unique microRNAs from good quality follicular fluids. So, and also the common microRNA showing some difference, significant difference. So for example, microRNA called miR9-1 showing 51 times expression more in the bad quality compared to the low quality. So this is also could be considered as a good marker that once you isolate the follicular fluids, you check this microRNA. If you found it highly expressed, so it means it could be the oocytes could be with a low quality oocyte. So that could be one of the application. And when we try to understand what this microRNA targeting, or what they are, which pathways or biological functions they related. So for example, the good quality follicular fluids microRNA, we found that they are targeting apoptosis process, for example, p53 signaling pathways related to apoptosis. And as I said, when microRNA is increased and targeting these pathways, it means this pathways is repressed or inhibited, because microRNA stops the expression of these genes related to apoptosis or genes related to this signaling pathway. So this is a good quality. It means the microRNA play a role to stop the apoptosis. And on the other side, you can see, for example, in the bad quality follicular fluids microRNA, they are targeting the oocyte meiosis itself pathway. So they are stopping the maturation of the oocyte. They are targeting estrogen signaling pathway, gap junction. So they targeting the opposite way of the good quality or the oocytes to compete or to ovulate a normal oocyte. So this is a good to understand that this microRNA could be used as a biomarkers just from follicular fluids, no need to touch the oocytes or the embryo. And it's coming out, you can see in the literature, a lot of paper talking about this characterization of microRNAs in follicular fluids and its relation with maturation of oocytes in porcine follicular fluids, in bovine, for example. Here they are interested in protein, not in microRNA. As I told you, different researchers are interested in different contents of these EVs, but all at the end will merge in one important biomarker. So if we know some microRNAs, some proteins, some lipids, they are interested or related to good quality, so we can select them all as a biomarker. Then the researchers start to think, what if now we know the function and we know the contents, so what we can get if we incubate these follicular fluids EVs isolated with the cells or with the oocytes during maturation. So this nice paper showing that these EVs can be uptaken by follicular cells, so they confirm this point. If you look to this picture, I hope that you can see clearly that each green dot, it's representing EVs or exosomes isolated from follicular fluids. When it's adding to the culture media, so it's taking in granulosa cells, in cumulus cells, in also oocytes. You can see here in the oocytes, and this is the zona pellucida, some particles can enter the oocytes through the zona pellucida, if we see it in the large photo here. Also, this is another work showing the same, that this green dots, it means the stained EVs, just to be sure or to understand or to confirm that the cells, the oocyte cells and cumulus cells, can take these EVs inside. So each blue dot, it means the DNA or the nucleus, and the surrounding is the cytoplasm. And you can see the green dots are in the cytoplasm clearly. They are enter the cells very easy. So this is very important to use it in the culture media and when we doing, for example, IVF or culturing embryos in the lab. So we can add these EVs to the media and see the effect. Again, also this is another work showing the green dots of EVs are inside the ooplasm or inside the oocytes, in the zona pellucida, in the cumulus cells. So they are distributed all over the oocyte. So of course they have effect on this oocyte. Also in the embryos. So when they culture the embryos, so this photo showing the cells of the embryo, and the green dots again is showing the EVs or the exosomes. They are entering the cells of the embryo. So the results of this research showing that when culturing the oocytes and adding EVs isolated from follicles of 3 to 6 millimeter, they increase the blastocyst rate or the development of the embryos significantly comparing to the EVs-free media. And also when this EVs adding to the culture media of the embryos itself, not the oocytes, also showing increase in the blastocyst rate. So this is very important application for the people interested in IVF field, that you can use different size, different kinds of EVs or exosomes to incubate with the culture media or the embryos to improve the quality and to improve also the development rate of these embryos. The second example is from seminal plasma. So EVs, as I told you, you can find it also in seminal plasma, and it's also from the size from 30 to 100 nanometer and 120 nanometer in diameter, and they are secreted from different cells of the reproductive tracts like epididymis or prostate gland from the males, and they found that they have a very important role in promoting the motility of the spermatozoa. So without EVs, the motility will be very low or could be affected hardly the motility and of course the fertility. So any dysfunction or disorder in the extracellular vesicles from seminal plasma could cause also infertility in males. This is one example of the results when they co-incubate also exosomes with the seminal plasma and they check the plasma membrane integrity. So plasma membrane integrity, it's very interesting that when they conserve this semen for 10 days, so still the plasma membrane integrity showing a high percentage compared to, for example, for the control. So control without exosomes, it was 35% plasma membrane integrity, but in a high concentration of exosomes showing like 67%. So it's a very big, and for 10 days conservation of the semen, fresh semen. So this is very also important for people doing artificial insemination and can preserve the semen for a long time. So adding of these exosomes to the semen can also improve the quality of the semen. This is a nice picture also with scanning electron microscope showing how these exosomes, these small particles, attach to the head of the sperm in porcine. So as you can see here, these all small white dots are exosomes or extracellular vesicles attached to the head of the sperm. And in this case, they release these contents inside the head of the sperm. And this affecting, as I told, the motility, the membrane integrity, the antioxidant capacity, and also the inhibit the premature capacitation under storage condition. And this group of researchers suggest that these exosomes, when they attach to the head of the sperm, they release some important proteins like AWN and PSP1 proteins. These proteins play an important role to inhibit the premature capacitation of the sperm until during the storage. So sometimes the problem when the sperm is stored for longer times, they start the premature capacitation, and of course this will affect the motility and the function of this sperm. So this is also a good application for using exosomes in the extender of semen. In a chicken also, there was a very recent paper published about the seminal EVs in a chicken in contrasted fertility. So they compare the fertile males and subfertile males. Interestingly, they found that the number of particles, which is less than 70 nanometer, was increased or highly increased in the fertile male compared to the subfertile male. So this is also one observation. And interestingly, when they also co-incubate these EVs with the chicken sperm, as you can see here in the photo, as I said, the green dots, it means EVs or exosomes. They are concentrated in the head, on the sperm head. This is the sperm head of the chicken male, and also in the intermediate piece of the sperm. So, but mainly they are concentrated in the sperm head to affect also this capacitation and the chromosomal reaction. And also they found that they increase the viability after the time of storage. Another example of this application of also to use it as a biomarker in superstimulatory response in cattle. So we know that most of cows in commercial health, they are producing 5 to 10 calves in their lifetime. But by using these technologies of superstimulation and superovulation, we can use these high genetically merits females to produce more embryos, and then these embryos could be recovered and transferred to recipients. Then we can increase the progeny coming out from this superior. But the main problem here is the variability in the superovulation response. Some cattle or some cows, when you superovulate them with FSH, for example, they respond very well and produce high number of oocytes. Others are respond very low. So this is also time consuming because we don't know which one will respond well and which one will respond low. So we have to superovulate all and then we can see. So this is time and of course money consuming. So what can we do if we can have some biomarkers that can say that this cow could respond well and this cow could respond low? So we also construct this experiment and we make a superovulation for a group of cows, and we collected bloods from these cows before superovulation and after superovulation. And we check also the response of these cows, some group showing high response with high number of follicles in the ovary, some others showing low number of follicles. So we have a blood samples from the same animal before and after superovulation, and we know this animal is highly respond or lowerly respond. Then we isolate the EVs and isolate the microRNA, and we also test the microRNA. This is some results. I will not show more details, but just to see that each kind of animal showing, or each type of respond animal showing different microRNA profile. And very interestingly, when we compare the high respond with low respond, we found some group of microRNA, for example, this one, near 206, was highly expressed in the low respond animals. So also this could be used as a biomarker, and these samples was isolated from the blood. So just you can isolate blood samples to check if this microRNA is present with high concentration. It means this animal could be low express or low respond to superstimulation, so don't need to stimulate them. But if you find other, for example, like this microRNA, you can use it as a biomarker, positive biomarker for good or highly respond animals. So this is one example. Another example also in cattle, in metabolic dysfunction, for example. So as you already know, the transition period from pregnancy to lactation is one of the most challenging periods. And there is a significant metabolic hormonal change occurring this time, and the body requires some increase in the intake of energy and proteins and so on. So we need to understand also, because if there is some fail in to successfully regulate this metabolic change, so this could lead to immunodysfunction or to increase the risk of metabolic and infection disease. So we need also biomarkers for this. That's why this paper, for example, this group, they found some important exosomes in the circulatory system, in the blood, related to high-risk metabolic dysfunction animals compared to the low-risk animals. And this is correlated, of course, to the health. So they found also exosomes, and they found here proteins related to the high-risk animals to this metabolic dysfunction, and also proteins specific for low risk. So they can be used also as a biomarkers. Another group also doing the same, but they collect from floccular fluids, and they see also that there is difference in microRNAs, and some microRNAs are related to the negative energy balance animals compared to the positive animals. So these markers could be also used as a biomarkers related to the ovarian function and metabolic disorders. So the last example here is from EVs from milk as potential also marker for cow health. So I will show you here also one example to collect from milk. So it's important to know that microRNA in the milk is more stable, and they can also be stable after heat treatment or during storage, and they are easily collecting because you collect milk, so you can just take sample from this milk and isolate these EVs. And they are highly resistant to RNA degradation, also for gut and digestion. So of course, this make it more bioavailable, and anyone can drink milk. So he drink also some EVs with microRNAs, with RNA that can also affect the function of the cells of the recipient organism. So this is very important to know what kind of EVs found in the milk, because this could affect really the organisms they are consuming this milk. This is very interesting, two papers published in 2 different years. For example, this one 2018, this one 2015, both searching for microRNA from exosomes in response to Staphylococcus aureus infection. So, and this is related to the mastitis disease. The very interesting finding that both papers found the same microRNAs, two microRNAs, MIR223 and MIR142.5p. Both are related to the mastitis disease and the bacterial infection. They found this microRNA in the EVs of the milk. So both papers suggesting that this could be a good biomarkers for infection. So if you find this in the milk, so this could indicate that there is infection with bacteria, or indication for maybe mastitis could be affect this cow. And also some other paper found the same microRNA in colostrum. So, and this could be related to the immune response or to increase immunity of the neonates or in the mammary gland after in postpartum. So, and because it will be susceptible for bacterial infection. So maybe this microRNA in EVs play a role in this immune response. So I hope that I didn't take too much time, but in conclusion, we can say that the field of EV biology is rapidly evolving and expanding too much in the last few years. And this would affect, of course, all disciplines of science, including animal production and reproduction, as I show you some examples. As I said, EVs can release from almost all cells in the body and can be found or detected in all body fluids. So that's make it easy to collect from any body that you are interested in, any body fluids. Also, we mentioned that they play a very important role. It's not dust, it's rapidly garbage, it's not a waste, but it's evolving and expanding communication system. So it's very important to understand this cell communication and to decode the communication to understand the function. If we do this, I think we will open up new horizons and breakthrough in this field of science and in biological science. However, there is some challenges in this field. For example, as you see in this photo, collecting the most important challenge. So, for example, the first challenge about the size diversity. So, what is the meaning of different size? Why they are different size of these EVs? Why they are not all in the same size? We need to understand this. Another challenge is the biogenesis. What is the difference in pathways and why some exosomes are generated in endoplasmic reticulum and endocytosis pathways, but microvesicles are just budding from the membrane. What makes this difference? We need also to see the cargo loading. What is the meaning? Is it specific that each particles contain specific microRNA or proteins or lipids, or it's random? So we don't know if the cells can select what kind of loading cargo inside the particles, or it's random. We need also to understand the target specificity. Is it specific that all particles released from one type of cells is going for a specific type of other cells, or it is also general? It can target any cells. So this is, we don't understand it well. We need also to see, because there is also a problem that EVs is like viruses. So there is a common, especially RNA viruses. They are having the same size, they have the same contents of RNA, and some reason papers showing that viruses using or can hijack the system of EVs to release its content. So especially in the last years that we see the COVID-19, so COVID-19 virus can use these particles to spread its contents of RNA to other cells. So this was one of the findings also. So we need to understand the relation between EVs and the viruses. Also, other a challenge is cell delivery. So how they are uptaken by the cells, how they enter the cells, and also the cargo-dependent processing, how they process inside the cells when they are entering, and how they affect the function of these cells in the health condition or disease condition. Is this EVs can spread the disease more, or can you be used also only as a biomarkers, or can be used as a therapeuticals? So all these challenges and all these questions, I think it's under investigation. And if we could answer this question, I think we will jump very huge steps in this field. And of course, it will affect all applications in all fields, especially also in animal science and in animal reproduction. So I hope that you get a benefit from that, and it was easy for you And I would like to thank you again all for your attention, and I will be happy to receive any question from you. Thank you so much.
Okay, thank you very much, Professor Ahmed. I think there are a lot of interesting information here that we got from Professor Ahmed, and yeah, we have already some question here in our chat room. First, maybe I can. Sorry, Prof, can you stop your share screen and then I
Yes, okay.
answer the question?
Moment. Yes.
Yeah. Yeah, this is the question from our chatting room, Prof. First from our participant from Bangladesh, Adnan. Maybe this is not related to our topic, but if you don't mind, please you just answer this question.
Yes,
First, can
yes.
you tell us the difference between MRA and MicroSA and so on?
Yes.
And then, yeah, I think
We can answer one by one.
Yeah, please.
Yeah, so yeah, it's a good question. So messenger RNA and relation with microarray or NGS. Microarray or NGS is a method to detect or to quantify the messenger RNA in the cells. So if you isolate, for example, RNA, and you'd like to see how many RNA copies of this specific gene, for example, you have to use microarray method or NGS. Of course, now NGS is much more important and used very rapidly more than microarray. Microarray, it was in the last 10 years, but now it's will be replaced by NGS, by next generation sequencing. So because next generation sequencing can quantify all RNA inside the cells, whatever this RNA is known or new genes or whatever, but you can see that NGS is more general and more cover of all kinds of RNA. So if you would like to quantify RNA or even microRNA, for example, as I showed in the presentation, we would like to see how many copies of this microRNA present in the cells or in the EVs. So we go to the next generation sequencing and we sequence all kind of RNA. So it's just methods for quantification and to detect the messenger RNA or microRNA. And the second, is there any relation between messenger RNA and micro? I think this is the same. As I told, this is microRNA is just microarray, and NGS is just method to detect and to quantify the messenger RNA and microRNAs.
Yeah, and then from Indonesia, Jilan.
Yeah. Based on what I know, extracellular vesicles have the ability to transfer their content to other cells, where they stimulate signaling activity, leading to phenotypic and change in recipient cell. If the EVs transfer malicious contents, how can it be resolved? Actually, this is one of the challenges that I mentioned at the end, that we not understand very well if these extracellular vesicles, how they are transferred to specific target cells, and how they affect also these cells. We know that there is effect, and we tried it, for example, when we co-incubate these EVs with cells or with oocytes or with embryos, they have effect. They change the development, they change the quality. But how they are changing this is a challenge. This is still a research question, and we need to understand if each type of extracellular vesicles have different function, or if they are all have the same function. Of course not. Each type has different function, and also not each type, but each released EVs from different cells have different functions. So cancer cells released EVs totally different than normal cells. Under heat stress, for example, cells will release EVs with different contents compared to the normal cells under normal conditions. So each physiological condition showing different EVs with different contents, and of course this could affect the target cells differently, but still we don't understand it clearly, so we need to do more research to answer this question.
Okay, and then
do
from
the EVs
Bangladesh,
have function in autoimmune disease? Actually, I cannot answer this because it's not in my field, but of course we need to search for that for people who work in medical science and to see if there's, but as I told, they have a role in immunity, of course. So there is a role of EVs in the immunoresponse and in immunity, but especially in disease of autoimmune response or autoimmune disease, I don't know, I didn't read actually about that, but of course we can search about it. So it's not in my field, so I cannot answer.
Okay, thank you, Prof. Ahmed. I think that's all from our chatting room. And yeah, we still have time, maybe for 10 minutes. If anyone has any question, please just to raise your hand, maybe. Yeah, and then you can directly to ask to Prof. Ahmed, please. Anyone, or all is clear? Oh.
I hope it's clear, or
yeah, I hope so. Yeah, but yeah, it's very interesting topic for Prof. Ahmed. Yeah, because it's quite very new for us, for me as well. Yeah, and then I think it's very interesting topic.
It's also new for all of us, actually, or most of us. So you will find now in research field, so most of the people are changing their view to this extracerebrovascular related field because it's a new one and it's very promising field actually. As I show that it's have a lot of application can be coming out, but first we need to understand it very well. It's not well understanding well, but we have some application at least on the research level.
Okay, thank you very much.
Okay,
Prof. Asep, please.
Okay,
you have any comment, maybe?
Dr. Ahmad is very nice presentation and also is good knowledge. Since maybe our work is not yet come to microRNA, yeah, you mentioned microRNA is control 100 gene, yeah, we still working with the gene, yeah, characterize gene expression or something like that. So in view of you, yeah, for the working with microRNA, we want to maybe to do a parallelly with the characterization of the gene and also with microRNA. So maybe to give the overview or understanding from from where we have to start, yeah, if we want to work with microRNA actually. So maybe this.
Okay, so to start, of course, you have to specify what is the question that you would like to answer, or what is the objective of the study. As I said, for example, microRNA play a role in regulating gene expression. So of course, as also mentioned, that one microRNA could regulate hundreds of genes. So expression of microRNA is very important. So first, you need to characterize which microRNAs you are interested in. So you can search in the databases. There is a lot of databases related to microRNAs that you can know each microRNA target which function that you are interested in. For example, you are interested in pathways related to oxidative stress response, for example. You will find a lot of microRNAs play a role in this pathway. If you are interested in a function related to reproduction, so or a specific thing in oocyte maturation, for example, there is a lot of microRNAs play a role in this. So you can specify first what kind of function or biological question you are looking for. Then you can target this microRNAs that's related to this function. You can start with characterize this microRNA expression to see if they are expressed high, low, or normal in your under your treatment or in your condition. You can also do another functional analysis of this microRNA. So we did also here some work related to inhibiting the specific microRNA. So we inhibit this microRNA in the oocytes and to see how this microRNA or how these oocytes could react when they miss this microRNA. So you can inhibit, there is chemicals now in the field or in the market. You can buy specific chemicals that can inhibit a specific microRNA. Then you can see the effect on the cells. Or you can just collect the microRNA and you can characterize. Another approach that you can do this NGS, next generation sequencing, just to discover what kind of microRNA, if you don't know any microRNA in your cells, you can just discover what kind of microRNAs are there. So you can just do sequencing. You can see a lot of microRNAs expressed, which microRNAs are very highly expressed, for example, in your cells, which are very low expressed. Then you can target specific microRNAs and you can understand the function of them. So there is a lot of approach that you can select, but first you need to put the question, what kind of question, and then you can find the related microRNAs in of this pathway or this question or biological field that you are interested in.
Okay. Okay. So this is interesting actually. Since sometime mostly we work with the transcriptome profile with microRNA or maybe NGS, but
Yes.
we are not overcome also, or maybe we are not characterize the microRNA. Since we found, for example, in our case, we work with the mid quality trait, and then we want to see the transcriptome profile in divergent phenotype, for example, a fatty acid composition or maybe a mid quality trait. And then we found, you mentioned about the pathway analysis. We found, for example, PPAR signaling pathway. And then we can come from there to characterize microRNA. It is like that?
Yes, it's possible. Now you have the specific pathway. So from this pathway, you can search for which microRNAs that targeting this pathway. So you will find, for example, like 10, 15, 20 microRNAs are related to this pathway or the genes inside this pathway. Then you can start to characterize or to focus only on these 10 or 20 microRNAs because they are very close or related to your pathway. So you can start it oppositely. That, as you said, you know the pathway, you know the genes that you are interested in. Then you can search for the opposite, which microRNA targeting these genes. Then you can target or to quantify or to see the expression of this microRNA, or as I said, to do some functional analysis to inhibit this microRNA and see the effect, whatever you want, but you already characterize which microRNA you are interested in. Some people who don't know any specific pathway, so he start from the beginning. So start to do sequencing for all microRNAs and see which microRNA are present and which are expressed high, which are expressed low, and then he can go further for analysis. But as you already know the pathway, so it will be much easier for you to select the specific microRNAs to this pathway.
Okay. Yeah. Now it's clear for me, and thanks a lot.
Great. Thank you.
Yes.
Okay,
Thank you.
great.
Bye-bye.
Okay, thank you, Pak Asep and Prof.
Thanks,
Ahmed. Well, okay. Is there still any question from participant? No. Okay, if there is no.
Ada, Pak.
Yes. This from Adnan again. Yeah, you can read in our chat room how to choose which pathway is appropriate, please.
Okay, so actually, for example, to choose a pathway, first you need to do this sequencing. For example, next generation sequencing, you will get a lot of genes are expressed or a lot of microRNA are expressed. You can see which specifically genes or which microRNA. Then you can go for some software. There is now a lot of tools, online tools and free tools that you can select. You can put, for example, the list of genes that you found, and then these tools can generate for you pathways analysis and can show you which group of genes are participate in this pathway. Or you can do the same with microRNAs. You have a lot of microRNAs or list of microRNAs expressed in your cells. Then you put them for specific tools or online softwares. They are distributing this microRNA in groups and saying that this group of microRNA, for example, targeting 10 pathways, this, and this. And this group of microRNA targeting another pathway. Then you can see which pathway are related to your cells. For example, if you are interested in blood cells, in liver cells, whatever, you can see the pathways or the function which is really related to this kind of cells. Then you can select this pathway and select the genes or microRNA which is targeting this pathway. Then you can go further for a specific or functional analysis after that. But to select from the beginning the pathway, you need to do a general analysis with sequencing generally, and then with using these online tools or softwares, you can distribute your genes in groups, and each group could target specific pathways. Then you can know your interesting pathway. So I hope this will answer the question.
Yeah. Adnan?
Thank you so much, sir.
Okay.
Yes, sir. Thank you so much.
You're welcome.
Okay. Is there any other question? I think it's, yeah, I hope it's clear, yeah, about our topic for today. But I'm sure, yeah, there is still any question maybe later
There's
on.
one question coming, I think.
Yeah.
Yeah, I would like to ask about
Yeah,
the
yes, it's
cycle of
coming again.
EVs and heat stress. Yeah. Country is a global warming that distributes the comfort zone, livestock well. This condition affect the number and the condition of extracerebral, for example, stress. It's very, very nice question. And we did actually one research related to heat stress. And it was very, very interesting that we just doing in the lab, for example, we isolate granulosa cells as an example or a model, and we stress these cells or put it under heat stress and compare with the normal cells without heat stress. Then we isolate the EVs from the culture media from both groups and compare. It was very interesting that number of EVs excreted in the media was different, was higher under heat stress. And also very interesting, the content of the microRNA contents also of these EVs was totally different. The most important application of that, when we take the EVs from the heat stress cells and co-incubated with normal cells, and after that we expose these normal cells to heat stress, we found that the heat stress cells respond much better and the viability was much better compared to the untreated cells. So that's give a very important message that the heat stress cells released EVs in the media that carry a message. This message warning the other cells, the normal cells, that take care, there is a heat stress, you have to manage yourself. So they can start to produce heat shock proteins, whatever, even they are not stressed. But after that, when we take these co-incubated cells and expose it to heat, they respond very well compared to the cells which is not have this message. So this was very clear, and we published this last year, and it was very interesting to show that heat stress affect the EVs, affect the content of the EVs, and also affect the other cells. So they communicate, cells are communicating to each other, like cells say that take care, there is a heat stress wave, so you have to respond. The other cells respond well and protect themselves before they are heat stressed. So this is very important. And of course, if we apply that for the whole body, not only the cells in the lab, this also could be a very interesting application that under heat stress, EVs can play a very important role either to protect the other cells or to use it as also biomarkers or diagnostic tools. So I think, yeah, and you will find a lot of papers and work about heat stress related to EVs. So, but I mentioned just what we already published in this area. So I hope this could also clarify your question.
Yeah, Fitri, do you have any comment, Fitri?
No, sir. Thank you.
Okay, thanks.
Thank you.
Okay. Any other question? Maybe for 3 minutes. Yeah, this from Reswati.
What kind of lab equipment that need to analyze EVs? In which country we can do this research that have completed? Actually, you can do, of course, this analysis in any country, but the lab equipments, as I said, it depends on the isolation method and the characterization method. So first, you need to isolate the EVs, and as I mentioned, there is a lot of methods, and now it's very easy to isolate EVs using kits. So you can just buy the kit, and you need centrifuge, vortex, and that's all. So it's very easy. If you have ultracentrifugation, it will be also much better. So you can use ultracentrifugation method to isolate the EVs, and it needs only centrifugation. So it's now easy to isolate. If we go for characterization, so you need electron microscope just to characterize your samples. You need Western blot analysis, protein analysis in regular lab of protein labs, and you need this nano tracking analysis. Usually, for example, here in our lab in Czech Republic, we don't have electron microscope, we don't have nano tracking analysis, but now companies doing this service for you. You just send the samples after isolation, they do the capture the photos of electron microscope, they do the nano tracking analysis, and we do also the Western blot here in our lab. After that, so you isolate and you characterize. So this is the main part of your that to be sure that you isolate a good concentration and you have a pure exosomes or EVs from your sample. Then you can go for whatever you want. You can do RNA isolation, and this is also need kits and centrifuge in the lab, and just to buy kits for isolation. You can do also PCR or real-time PCR just to specify specific genes or specific microRNAs. So you need real-time PCR. Or if you would like to do next generation sequencing, you can send your RNA isolated or isolated samples to companies to get this thing. So here, for example, we don't have also next generation sequencer. So we send our samples after isolation to companies, and they do this sequencing and send us the data. So it's not complicated. If you have a normal lab, you just need centrifuge, PCR, the basic equipments, and you buy kits for isolation, and that's all. Then you can send your samples in any country just to do the sequencing, for example, or some specific characterization. So it is not complicated. Let's make it more spreading that the research of this EVs is more than because any lab can do this analysis. It's not, of course, you need money because it's a little bit costly to buy kits, for example, for isolation or to do the next generation sequencing. This is a costly part of the technology. Yeah, so it's not complicated. You can do it in any place. Just if you have basic equipment in your lab and you have money to get these kits and characterization methods and also next generation sequence.
Okay, I hope it's clear, yeah, Reswati, yeah. And then this this is last question, maybe, sir, Prof, from Murshid Hassan. Can you solve the challenge by using probiotic in dairy cow?
It's hard, or of course, to know. We need to try to solve these challenges using probiotics. Maybe it could solve the challenges, but the challenge is to understand the normal behavior or the normal situation in the natural, in the animals. So without any treatments, without any disease, without anything, we need to understand how the animal or how the cells are releasing these exosomes, how they are captured by others, because of course they have physiological function in normal conditions. So in all bodies, they release every time, every second, you will find exosomes in the blood, in the fluids. So they have a function. So without any using any other outside source, we need to understand first the natural, how the nature or how the animals or how the body deal with these exosomes, how they release them, how they uptake them with other cells, how they affect the function of these cells. Then after that, we could go with other application like probiotics, for example, to see how these probiotics could improve, for example, the function of these exosomes, or how they can improve the uptaking of these exosomes and so on. So I think first we need to understand the natural situation, and then we could go with any other treatment or any other outside effects.
Okay, I think everything is clear, yeah.
Thank you.
Thank you very much, Dr. Ahmed sir. I have learned a lot of things, the biogenesis and other things, the structure, functions, and application. Latest information regarding EVS. Thank you very much for your nice presentation, sir.
Thanks for your comments. Thank you.
Okay, now coming to the end of our session for this class, and yeah, before we close for this session, let me invite Professor Irma. Are you there, Prof. Irma? Vice Dean of Faculty of Animal Science.
Yeah.
Yeah, Prof. Irma will help. Yeah. Yeah, this is the certificate of appreciation and will be given with by Prof. Irma. Prof. Irma, please, time is yours.
Thank you very much, Professor Ahmed. This is very interesting lecture. And I'm very interested with some topics about microRNA, specifically to detect the microbes in some materials. For example, to detect the mastitis in the cow, and then maybe for the probiotic, how the beneficial probiotic in our body and human body, and so on. And this is my my field also in probiotic and microbiology. So I am very interested to hear the lecture, and thank you very much for your attending to give and to deliver the interesting lecture today. I am Vice Dean of Animal Science Faculty at IPB University. Very thank of your attending today, and thank you very much. Thank you very much, Prof.
Thank you, Professor. Thank you for these nice words and for inviting me for this. It was a great pleasure for me actually to join, and I hope that I can join also in the next courses or next year, hopefully. So it will be my pleasure. Thank you so much.
Okay, thank you so much, Prof. Irma. And yeah, ladies and gentlemen, and now it's finished for our class for today. And again, thank you very much, Prof. Ahmed Gad. I hope we still have a contact for the future, maybe next summer courses for next years. Yeah.
Of course,
thank you very much again for joining us.
Thank you so much. Thank you. Thanks for all audience for their interest and for their attention. Thank you.
Thank you, Prof. Ahmed. Barakallah. Jazakumullah khair.
Shukran jazilan. Jazakumullah khairan.
Okay, thank you. And then, yeah, and feel free, Prof, if you want to leave this meeting because we still have any, I mean.
Okay, great. Yeah, because I have to have another things in the lab,
yes.
So I have to leave now. So
okay.
Excuse me. Yeah. So
okay.
enjoy your course and have a nice day.
Before you leave, please,
yes.
We want to take a picture together here. Please,
yes,
everyone, if you have please to on your camera, and then the committee, please.
Pak Arman atau Pak Dimas ya, or Pak Dimas.
Okay, thank you very much.
Thank
Again,
you.
thank you very much for
Thank
attending.
you. Thank you, Asif. Thank you.
Okay,
have a nice day. Bye.
Yeah.
Bye.
Okay, and now coming to the next session is, I forgot about the schedule, but I think we will have, yeah, video, yeah, from the committee. And today the video is about Java Island, I think, yeah. Please, yeah, please, you can start the video.
A land of myth with ancient temples stretched far and wide as the legacy of historical greatness. Like the ones found in this 15th century palace of Cherabon. Take the train, let it take you back in time, and enjoy the timeless feel of a magic ride. Here, every tale is expressed by genuine human relationship and by the deft touch and tender care of artistry. The great beauty of batik has a life of its own in this land. Time-honored values are preserved in body and soul. This is where a mask dance brings us wisdom about life. And the goat fighting demonstrates the meaning of survival. Bamboo has a special place in everyone's heart. Its melody is nothing but an infectious charm. The charm that echoes throughout the world. The plant is everywhere. It is a part of life. It unites the people with the mother of nature. In a sense, bamboo has become the epitome of harmony. Meanwhile, in the South Coast, witness the existence of a sustainable life. A life reserved for the generations to come. In this wonderful hidden paradise, if you love water adventures, you're in heaven. From the ocean up to the river, nothing is off limits. Adrenaline rush knows no boundaries. It gives you the moments to live for. Just outside the city, there's a collection of wild animals from all over the world. Social understanding is developed. Trust is established. Intimacy is formed. In the capital, tradition comes in style. Urban festivals frequently grace the streets. Local dishes and international menus are presented. In the warmth and friendly manner Bandung is known for. Fashion and lifestyle have long been the synonym of the city. It is a shopping nirvana. The lush and beautiful landscape offers only the best of serenity. From the five-star hotel's luxury to the intimate lodge's tranquility. You can always take a journey to the mountain and walk down to the crater. It also has the most extensive botanical garden in the country. It's a perfect choice whenever you want a place of solitude. Your time and effort will be immensely rewarded by the spectacular beauty of West Java. Wonderful Indonesia.
A wondrous civilization. Disposed to tranquility. with scenic landscapes bordered by the sea. Here, mythologies become principle. And religions create the cornerstones of life. It is contained in the arts and crafts, where celestial ceremonies become part of popular culture. Legacies of glorious traditions, where folktales are regarded as guidebooks to perfection, honed over centuries, yet possible to experience within a few hours. Experiences you can take home. Here, art preservation is a tourist attraction. History is a living monument. Even shopping is meaningful. See world-famous resorts compete with local means of accommodation. Enjoy natural kindness. Or the excitement of exploring nature. With endless stories to tell and never-ending feasts. Never-ending possibilities to get more out of life and journeys. Yogyakarta. Wonderful. The mountainous region of East Java. Where Majapahit once stood. Largest ancient kingdom. Changing and opening slowly over hundreds of years. A historical city on the move. A celebration of remarkable heritage, grandeur of the past, wondrous ancient architecture, and the outstanding universal values behind, where art absorbs the audience into colorful drama. Still, the exotic mesmerizes. Extraordinary feasts exhilarate. And bring us back to the land of the ancient mountains. of outdoor adventures. In exceptional panoramic nature. Exciting variety. This is a land of historical significance, where the old days are celebrated. Local cuisine is cherished. Beyond the city, preparing for a thrilling ride on the vast sea of sand in Bromo, East Java. Wonderful Indonesia.
Okay, do we have? Okay, please. Okay, thank you very much, Pak Arifin. Yeah, ladies and gentlemen, we still have time, for break, more or less for 8 minutes, and then afterward, in 3 o'clock Indonesian time, we will start for the session 10, with Dr. Salome Mikuse. I think she is already joined with us. So, we have a break, more or less for 5 minutes, and afterward we will start for the 10 class. Okay, thank you and see you around. Okay, thank you, Pak Arifin. And now it's already 3 o'clock in my time. So I think everybody should join with us from now. And yeah, as you may know that for the next session is presentation from, the lecture from Dr. Salome Mikuse. Let we greet her. Hello, Dr. Mikuse. Are you there?
Hello.
Hello.
I'm not sure if I can pronounce your name correctly.
Yeah, just call me Bai.
Haji?
Bai, it's okay.
Bai,
Yes.
okay.
Yeah.
Yes, I'm in. The network may be a bit disturbing, but I'll try my best.
Okay, no problem.
Yes.
As long as your voice is can hear by us.
I wanted to show my face a little bit, and probably I do a short video, not for long.
Okay, please.
The internet may not be stable. I don't know if you're able to see me. Hello?
Yes, we already see you. Hello?
Yes.
Yes, okay. I think good morning, in your time.
Yes, it's still morning. It's 11 o'clock.
It's 11 o'clock. Okay. Thank
Yes.
you, Dr. Mikuse, to joining us. And before I give your the screen for you, let me share my screen to introduce our speaker for this session. Yeah, I hope everybody can see this slide. Yeah, her name is Dr. Salome Atino Mikose. Sorry for wrong pronunciation, maybe. And she comes from Department of Agricultural Resource Management, University of Embu, Kenya. Yeah, this is her email address. Maybe some of you who want to contact her maybe later on. Okay, and this is the education background of Dr. Mikuse. Dr. Mikuse finished her BSc and MSc program in University of Egerton or Egerton University in Kenya. Meanwhile, her PhD program has conducted in Wageningen University and Research, the Netherlands, in APS program, I think, in Zodiac building. Working experience, from 2020, as you see in the screen, he became a lecturer in University of Embu, Kenya. Before that, as a tutorial fellow, also in University of Embu, Kenya, and so on and so on. You can see as well here. Yeah, this is some certification and also short courses that she took during maybe in PhD program and also in another program. There are also a lot of certificate and short courses that she completed. And then she also has several publication, as you see here in the screen, from in journal, the Fritz journal. She has 5 publication, and also there are many abstract in conference proceeding. In this lecturer 10, Dr. Salome Atino Mikuse, or I can call you Dr. Mikuse maybe, will have a topic about the domestic animal genetic resources and their livelihood. Okay, Dr. Mikuse, I can stop my screen, and then please, if you want to share your screen, and the screen is yours.
Thank you, Bai. I'm grateful to have this opportunity to share with you some or give a lecture on animal genetic resources, in particularly in Kenya, and how they contribute to the livelihood. I just wanted to prepare you. We may have some disruptions regarding the internet, particularly if the power goes off and it goes with the internet. So I'll have to then now switch to link via a hotspot or a modem. So you will just be patient for a little bit. If that happens, then I'll be back. Then I also, sometime you may hear a little bit of a background noise because I'm in a shared office. So kindly bear with me, but in all, I hope for the best. So I'll try to share my screen. I have 3 slides or they're in 3 sections, not 3 slides, but they're in 3 sections. So I'll start with the first one, and just a minute. Yeah, I think that is now clearer enough. So I'll talk about the domestic animal genetic resources and their livelihoods in Kenya. So I don't know how, okay, I think, yes, I can do this. But so as a way of introduction, the animal genetic resources are basically the animal species, breeds or strains, and their wild relatives that are reared for their economic, scientific, and cultural interests to mankind in terms of food and agricultural production for present or the future generations. So if you do a small Google search for the animal genetic resources in Kenya, you're likely to come up with all sorts of these beautiful animals. Then this is because the climatic conditions in Kenya is very diverse. We have what we call agricultural ecological zones ranging all the way from 1, which is the mountain areas, to 7, which is the hot and dry climate or the deserts. So from this picture here, majority, over 80% of Kenya falls in what we call arid and semi-arid areas. So, and then we have less than 20%, which is in the highlands and lowlands where we do agricultural production, but in the arid and semi-arid areas, we keep a lot of livestock, especially in the rangelands, ranching, and also in the pastoral systems. And this is like the sole source of livelihoods for the people who stay in this environment. So in Kenya, we have several species of animals that are domesticated. So for the population of livestock, we have about 19 million cattle. That is according to the latest census that was done. About 28 million goats. These are will comprise various breeds, as I will show you later. 19 million sheep, 3 million camels, 45 million domestic birds, about 2 million donkeys. And then we also have companion, game, and aquatic animals, which their numbers are undetermined. At the same time, we also have what we call non-conventional cattle, rather livestock. I will discuss that further. So if you compare Kenya to our neighbors who are found in Uganda and Tanzania, this table gives a comparison of the animals. I don't know if I'm audible enough or I need to add the volume.
Hello?
I don't know if I'm audible enough or I need to increase the volume.
Yes, it's enough. Yeah, it's clear enough for me.
Okay. Then I continue. So I just wanted to highlight that from this table, it's clear that especially the goats and the sheep, the small ruminants, we are really having a relatively higher population when we compare with our neighbors, the Uganda and Tanzania. Okay, so I said we have, when we talk of cattle, we have several breeds. So we have the exotic breeds. These are the breeds that were imported from the temperate countries, and they were brought by the colonialists during the 1900s. And then, so we have dairy breeds and the dual purpose breeds. And then we also have, we used to have beef breeds, but at the moment, there are really not so many because of the disadvantage of the climatic conditions. Then we have several indigenous